Welcome everybody to today's Penn AI Tech webinar. Penn AI Tech is the Penn Artificial Intelligence and Technology Collaboratory for Healthy Aging, an initiative funded by the National Institute on Aging. Today, we're very excited to have with us Dr. Nathan Price as our speaker. Dr. Price is professor and co-director of the Center for Human Healthspan at the Bach Institute for Research on Aging, and is also Chief Scientific Officer of Thorne, a science-driven wellness company that serves more than 5 million customers and 47,000 healthcare practitioners. Previously, he was CEO of Onegevity, an AI health intelligence company that merged with Thorne in 2021, and professor and associate director of the Institute for Systems Biology in Seattle for a decade. He's co-author with biotechnology pioneer Lee Hood of a 2023 best-selling book, The Age of Scientific Wellness, published by Harvard University Press. In 2019, he was named one of the 10 emerging leaders in health and medicine by the National Academy of Medicine, and in 2021, he was appointed to the Board of Life Sciences of the National Academies of Sciences, Engineering, and Medicine. He's also a fellow of the American Institute for Medical and Biological Engineering and Camille Dreyfus Teacher-Scholar, and was the 2023 recipient of the Alexander Mildred Seelig Award for Science from the American Nutrition Association. We're very happy to have Dr. Price with us. I want to remind our attendees that if you have questions for our speaker, you can use the Q&A feature at the bottom of your Zoom screen, and we'll get to the Q&A session after the talk. So with that, Dr. Price, thank you again for joining us. I'll hand it over to you.
Great. Thank you, George, for that kind introduction. It's great to be together with everyone. I'm hoping to aim this a fair bit shorter than the hour, so we have some time for back and forth and discussion at the end, I hope as well. Let me just pull up my slide deck here. Okay. So, yeah, pleasure to talk with everyone. And so today I want to talk about what Lee Hood and I have called the age of scientific wellness, but basically what I think so many of us are so excited about right now, which is the opportunities to try and accelerate health span in a major way in the years and decades that we're going into now. So we're all here at a seminar that is around AI. And this is one of the things that I know is on a lot of our minds all the time, which is kind of our point in history at the moment, as AI systems are going from having been dreams into realities. Lots of limitations still, lots of promise, lots of things that we could dive into about what current systems can and can't do and what they may or may not be able to do. But it is an incredible moment for us to really think about what are the things that used to be hard that are now easy? What are the new areas of possibility that exist that didn't exist before? And how do we translate that? And one of my favorite phrases actually is from Stu Kaufman in one of his books that I really like, which is this notion of what is the adjacent possible. You may have heard him talk about this. I really love that notion, which is, there's possibilities, there's the possibility space that we all think about in research all the time. And the adjacent possible is like what has just become possible for us to do because of new discoveries, right? You couldn't go back 100 years and invent AI, had to happen now. It has to have pieces that are in place. And so this is one of the things that I think all of us are thinking about a lot right now, which is how do we do things in new and different ways? I would also argue, as I think would be true for a lot of the kind of people who would want to be on this kind of a webinar, which is that the abundance of health would be the greatest application of AI to humanity. And I think it's one of the things that we're all hoping to get, right? A major acceleration in our ability to increase health span, to combat aging, to not die of cancers, to avoid neurodegenerative diseases, et cetera, et cetera, et cetera, right? And so this is one of the things that we really want to push forward as much as possible. So I'm going to give my definition of aging, or at least one that we use quite a lot at the buck. Other people use it as well. I don't want to call it my definition, but anyway, the definition I like the best, I'll say it that way, about what is aging. This actually, I think about this. I don't know why this stuck with me, but when I was an undergraduate in chemical engineering, I remember there was a talk a lecture that was given, and there was an interesting fact and that came up, which was that light bulbs don't age. And I always thought that was interesting. And what was meant by that question? So these are like the incandescent light bulbs where you've got a little copper filament or something like this. And what was meant by that is that a light bulb is just as likely to blow out on day one as it is on day 35,001, because that filament is not actually wearing out by the electrons passing through it. Light bulbs burst because of a surge in the electrical grid, so it's sort of unrelated to uh, to the passage of time for them. So we talk about them not aging. And so as humans, as we think about it here, then, what I think is the most practical definition of aging, or at least the one that I favor, is just this increased slope, right? Which is as we get older year over year, our odds of getting all these different diseases, as we know, goes up. And so if we ever talk about, removing aging or solving aging or reducing aging or anything like this, to me, it's really about the slope of that line just getting pushed down, right? And you can imagine that if that line were ever flat, that would mean that we're not aging in a sense because our odds of having any of these terrible things happening are not going up at the rate that they are. And I like that kind of a definition because it's measurable, very actionable. There's lots of different ways that you can go about trying to alter that over time. So the other thing that we think about a lot, obviously, is this relationship between lifespan and health spans. One of the things that we have to be really mindful of in the United States in particular, because at least according to a number of metrics, we have the largest gap amongst developed worlds developed nations, excuse me, between I'm watching too much sci-fi lately. Anyway, between developed nations, that between lifespan and health span, and we have this gap that is a problem, and all of us experience that to some degree, larger or shorter in our lives. And so, one of the things that I've found really interesting when I first became a longevity researcher is if you're just chatting with people in the public, that you get a kind of mixed response to it. Some people love it. They think this is fabulous that you're doing this. I was quite shocked when I found out some people sort of hated this, or they felt very negatively towards that notion of living longer. And in thinking about that, I think there are definitely reasons for why people think that way. And one of these, which I think has been put really well, in fact, I was going up to give a talk on a similar topic as today at RPI a few years ago, and just driving up, and just by coincidence, I've been catching up on a book. It's called The Moral Epistles. It's 2,000 years old. It's by one of the Stoics. But just by coincidence, the next one that popped up was on longevity. And it was interesting to think and hear what someone 2,000 years ago was thinking about longevity. And he put it this way, which I really liked, which is, "For it makes a great deal of difference whether a man is lengthening his life or his death." And to me, that was so much the essence of, are you increasing health span or lifespan? And so when I come back to like that difference in reaction that people have to being a longevity researcher, I think this is really what is in their minds, which is, if you're imagining that you're extending your life, that period of vitality and health, and as you're enjoying the being alive, that is very potent to people. If, on the other hand, you're imagining the extension of death, which is the extension of that oftentimes terrible period at the end of life that people go through, that can be a scary proposition for people. And so there is a lot of difference when they think about, those of us who are longevity researchers, are we extending life or are we extending that period of death? And so I think all of us would be very strongly on the standpoint of the goal is to extend health span, to extend that period of vitality and health for as long as we possibly can. So the way that I come into this, or at least think about this, is around a concept that Lee Hood and I have called scientific wellness, call it precision health, a number of different terms. But basically, and one of the things that we wrote about in this in our book here is that is basically an argument that fundamentally the way that we've organized biomedicine and healthcare and so forth is too focused on late stage disease and not focused enough on what we can do early on to advance and promote and preserve and enhance health during the course of life. This is manifested in many different ways, right? We organize the National Institutes of Health into institutes for disease. There's a lot of work on biomarkers. There's a lot of work on drugs. These are all good things, by the way, and I've done a lot of that kind of work as well. But it has been limiting in a sense. At least I would argue that. So one of the things that is interesting to contemplate is the foundations of medicine. So when we have, or at least the foundations of kind of modern medicine. So this began really last century with the Flexner Report, bringing science into medicine, and it was really transformative. If you think about what people were dying of in the early 1900s, there's kind of morbid statistics. One of them is every third coffin was filled with the body of a child who had died from an infectious disease. And so we would have been losing, all those of us in the audience here that have children, right? A bunch of them would have died. They'd have these terrible diseases and so forth. And so when medicine really brought science in, and a lot of public health measures, vaccinations, antibiotics, clean water, all this stuff, the organization of medicine under a scientific framework, we changed the world, or at least the people that were involved in that changed the world in an incredible way, where we don't fear, for the most part, the kind of diseases that were absolutely a plague for us 100 years ago. Now, though, if we fast forward and we think about how successful have we been against chronic disease, and the answer is way, way, way less have we been successful against chronic diseases, against cancers, against dementias, against all these kind of things. Improvements have been made. We've made progress. There's been things, but we can't yet look and say that we've relegated to the dustbin of history any of these chronic diseases the way that we have infectious diseases. In fact, I spent 4 hours the day before the holidays chatting with Larry Brilliant, if you know him, who was actually the person that led the eradication of smallpox from the earth several decades ago now. But pretty incredible what has happened. And so part of the argument here then is that there are pressures that we're driven towards, both economic and practical, for studying disease in its late stages. It's an easier place to get a p-value. It's an easier place to get an R1. It's more reimbursable. And in fact, one of the things that's really interesting in our system from an economic perspective is we actually place a lot of economic value on years of sick life and no value on years of healthy life. We can maybe argue about that a little bit in the question and answer, but it's interesting because if you have a new drug, right, that can be very expensive, and they often are, right? The new Alzheimer's drugs, right, $25,000, $40,000 a year. But if you want to do a program on prevention, it always has a particular criteria, which is that it must save money. And so that is an interesting way in which we bias a system such that we will pay for a lot of money for preservation of years of sick life, less so for years of healthy life. There's psychological reasons for that too. But anyway, an interesting way to sort of think about, as we're organized, how do we structure so that we can focus more on some of these early interventions. The other big issue, and this was one that we really set out to try to get into more, and it's become way, way better in the last 10, 15 years, is that we haven't had a lot of data, really deep data during wellness in the way that we have had a lot of data on the study of disease, of case control, disease designs, and things of that nature. A panel I was on right before the beginning of the pandemic with the chairman emeritus of Harvard Medical School, which was really interesting. And he put this in a way that I really loved, which is healthcare is the only industry that does not study its own gold standard, which is wellness. And I think that has actually started to change quite significantly in the last few years. And indeed, I'll show that here, that this focus has really begun to change. And we're starting to see a lot of interest in this. We've seen the emergence of numerous companies in this space. I wrote this article for Scientific American in 2022 on evidence-based wellness emerges as an industry. Going through a lot of these companies, right? We've seen the emergence of wearables. We're seeing more and more people get deeper measurements on themselves. We've seen the rise of the biohacking community. We've seen a lot of these things. And here on the left, what I want to highlight, which was a report that I'm very interested in here from Deloitte, and again, this is from just a few years ago, where they forecasted that while current healthcare spends almost all of its money on treating sickness, that they're projecting that by 2040, that the shift will be that there'll be more actual investment in promoting and restoring health than in late-stage treatment of sickness. So you can believe this or argue with this, but there was kind of a threshold moment where you had one of these big, business forecasting companies actually coming in and agreeing with this premise of that we are actually seeing a shift. And in fact, we're seeing in the wellness industry a pretty big boom right now in terms of adoption and focus and trying to build systems that are more aligned with that. And there's a huge interest in this in the public. And so it's something that has been gaining steam, I would say, over the last decade pretty strongly. So we can think about this in terms of how this would manifest in a particular disease or a particular area of health. Wrote this piece with Lee Hood a couple of years ago on kind of around Alzheimer's disease and looking at that. And there's different estimates on this. Excuse me. There's different estimates, but we spent at least tens of billions of dollars, maybe hundreds of billions of dollars collectively on trying to deal with Alzheimer's from a late stage standpoint, focused a lot around the elimination of amyloid and things of that nature. And that has been not terribly successful. There's a couple approved drugs. You get a little bit of benefit. You get a little bit of slowdown to the rate of decline at very high cost. Now, conversely, there's actually been some trials, including the Pointer trial that was successfully done here in the United States, and reports on that a couple months ago. But started with the Finger trial. It's antecedent, and basically, there are different lifestyle interventions that you can do aimed at prevention, and you can get years of benefit at very low cost. And so there's this big sort of discrepancy between cost benefit analysis as you think about this as from a prevention standpoint versus a late stage care standpoint. So I'm going to spend just a moment here and talk about this a little bit. So we had done some work with a partner a few years ago looking into this, and there's different ways that you can evaluate Alzheimer's. So I just want to make a point around how we might think about this differently from a prevention standpoint. And so one of the things that you can look at is an understanding of what might drive this disease. And this is one of the things that we're really interested in at the Buck as well, which is not just thinking about genetic risk and do you have higher or lower risk for a disease, but when does that genetic risk manifest? And what mechanism of aging is it that made you now susceptible to that? So an example on Alzheimer's is the very famous ApoE gene, ApoE4 giving higher risk for Alzheimer's, ApoE3 being neutral, ApoE2 giving lower risk. Um, and I have one copy of ApoE4, not terribly uncommon. A lot of us do. But basically, that variant doesn't really matter to dementia in your 20s, your 30s, your 40s, usually your 60s. But as you get up in your upper 60s, 70s, 80s, it can make the difference between you, right, remembering the names of your children and not. And we all know the terrible consequences as Alzheimer's gets going. So if we think about kind of the needs if we flip this around in terms of a health status, Alzheimer's, if we think about brain health, so we started to study, what mattered in terms of keeping your brain alive. And so one of the things that we know, right, is that your brain is a huge energy hog. It takes 20% of it's 20% of your body's energy. It is 2% of biomass, where it's a huge energy hog, and you have to feed energy up to it all the time. So one of the things that comes out of the literature is that as you get older, right, your ability to perfuse oxygen into your brain goes down. And in some work we did with a company some whiles ago led by Tom Patterson, we had analyzed like these coupled ordinary differential equation models in the brain. And it turns out that if you want to keep energy generation in neurons high during low oxygen conditions, you want to keep cholesterol levels and astrocytes low. There's a coupling between them through the lactate shuttle. But the point I want to make is that ApoE plays a role in the transport of cholesterol out of astrocytes. ApoE4 does it really slowly, so that efficiency is poor. ApoE2 does it fast, so that energy efficiency is high. And that difference in efficiency is highly predictive of the rates of Alzheimer's disease across the population for all the different genotypes. So the point I want to make here, which is a larger point, I'm maybe giving a little too much background to get to that point, is if you switch then from thinking about this from a how do I maintain the brain's health instead of how do I diagnose a disease, you move away from like amyloid and hitting amyloid, which we could talk about more. And you move more to what is that mechanism of aging that matters? And in this case, it may be that mechanism of aging that matters is this excess metabolic capacity you have to keep your neurons alive that's ticking down as you get older. Because as soon as you become near threshold, that efficiency difference from your ApoE genetics all of a sudden becomes life or death. Whereas for most of your life, while you have that excess capacity, it wouldn't be a problem, not an issue. And so what I want to point out here is that if we think about it today, we have no mechanism for measuring today directly that excess metabolic capacity of the brain, how much it's decreasing, how close you are to that transition, what you can do to protect it. There are certain things you can do, at least that seem like they would be protective. Sauna use is one, right? We know from epidemiological studies that that is quite protective to dementia and so forth. But there are things we could imagine, but you would go about the process of going at a chronic disease very differently if you're thinking about it from the standpoint of what are the resilience aspects, what are the health needs, and where are we at in terms of making those better to keep you from transitioning in the first place. So that's the kind of thing that we're really thinking about a lot these days. All right, so to move forward, so we have as we were thinking about scientific wellness, and we started this about 12, 13 years ago, wanted to start generating a lot more deep data sets for that beginning during wellness. And so we recruited through first something called the Pioneer 100, which is this pilot project we did, and then through a company that we had co-founded, got that up to about 5,000 people, and basically just started collecting a lot of data. So this was whole genome sequencing on lots of people, doing about 1,200 measurements, labs out of the blood, clinical chemistries, metabolites, proteins, gut microbiome. We're doing these all 2 to 3 times a year, depending on how far in the program we were, and then continual self-tracking through, at the time it was Fitbits and things like that, but basically wearables, database of actionable possibilities so that people could utilize information to try to improve their health, and then created a lot of deep, dense data. And so this helped us move to kind of a practical standpoint of what kind of things could we learn by just generating a lot of dense data off of a population of some thousands of people. And I'll walk through a few vignettes around that now. So here we have microbiome, and I'll do it in sections. So I'll talk a little bit first around microbiome and metabolism. So one of the things that we learned was that as we looked at the microbiomes people in these cohort, that around age 50 or so, we started to see an increase in, it was kind of an odd score that we developed, but something that we called uniqueness. Basically, it has to do with how different you are from every other person in the cohort. And what we saw was that people's microbiome started to drift further and further away from each other. We quantified that. This was supported by a Catalyst Award from the from the National Academy of Medicine. And then we published this in Nature Metabolism. And what it showed here that was interesting, though, is that when we looked at this uniqueness score in an older population, in this case, a validation cohort called Mr. Oss, basically what we saw was that this higher microbiome uniqueness was strongly associated with higher walking speed, with being composite healthy. I guess here's the discovery in the validation cohort, being composite healthy, and so forth, having like higher health. And that diversity, in fact, was not statistically significant, which was interesting. And that score was, in fact, predictive of all-cause mortality over the next 4 years in this elderly cohort. One of the interesting things there was that there was a divergence in terms of the species in the microbiome, but we did see a lot of consistencies in terms of the positive and negative health aspects of the metabolites that were derived from the microbiome. So you saw much more of the positive metabolites like indoles and p-cressyl sulfate, and then there were other toxins that would come up that were associated with negative health, like TMAO, which is a known cardiovascular risk factor, and things of that nature. So there is this tight coupling actually between the microbiome and what you see in your blood. About 18% of the metabolites in the blood is estimated to come from, or at least be co-produced by the microbiome. And that's in fact something that we delved into here, which is that we tried to see if we could predict things like the diversity of the microbiome out of the blood. And it turned out that you could leverage or you could utilize the blood and capture about half of the variance of a signal associated with that just in the blood. And in fact, when we looked at this across a wide range of different conditions, the beginning of the alphabet, I guess, is shown here just to zoom in on it. But basically, what we saw was that as you had shifting in the diversity of the microbiome, we saw similar shifts in the predictions from the metabolome. So it showed that we could predict microbiome diversity pretty reasonably from the metabolites in the blood. And in fact, we found this to be quite strong across all the conditions we tested, except for one, which was in extreme obesity. So if someone had a BMI above 35, what the World Health Organization would call obesity type 2 or 3, here what we went for is, instead of capturing about half of the variance, it literally went to basically zero here. This is for the number one metabolite. And so essentially what we saw was this kind of obliteration of this relationship between the metabolome and the microbiome in extreme obesity, which we think is related to the high degree of inflammation and so forth, although we're not really sure. We don't have direct evidence for exactly what would drive that. And that's all published in Nature Biotechnology in 2019. Another thing that we looked at in these data that were quite interesting was what was predictive at baseline of if someone would go on to lose weight over the next few, over their time in the program, which could be up to a few years in the future. And what was surprising is that once we controlled for BMI, because BMI itself was, of course, predictive, but once we controlled for BMI, I was surprised by this. We got no additional signal out of the metabolites. I really thought we would, but we did not see that. Same thing in proteins. Again, somewhat surprising to me. Asking them what they ate trended a little bit higher, but in fact was also not predictive, statistically significant in our cohort. The only thing that were the genes of the microbiome, which was quite interesting. There were two things that really mattered. One was the growth rate of the microbiome. Maybe every calorie you eat is a calorie for you or your microbiome. Maybe you get a little bit of a benefit if it's churning a little bit more. But the main thing that made a difference was if you had gene content that suggested that you would more likely break down complex carbohydrates, eat a sweet potato or something, you would more likely break down carbohydrates into short chain fatty acids. Those people went on to lose weight at a much higher rate. If their microbiome had genes that suggested they would more preferentially break that down into simple sugars, those people had a harder aspect to lose weight. One of the interesting things is that those short chain fatty acids that are helpful, one of which is butyrate. For what it's worth, butyrate is a GLP-1 agonist. So an interesting connection there potentially. We also looked a lot at the metabolites themselves, and we did a regression, very much like you would do a biological age calculation. We did a biological BMI calculation. This is published in Nature Medicine in 2023. And basically what it did is show BMI is, of course, a very coarse measure. It's really more of a population measure than a very useful individual measure, right? Because if you're very muscular, right, your BMI is high, and that's not necessarily a sign of bad health. In the paper, we estimated that BMI pretty grossly misclassifies about 30% of people, and you could get much better results, we showed in this paper, in terms of the matching to health by looking at multiomics on a biological BMI type basis. If you're interested in the details, it is in the Nature Medicine paper. We then connected this back to the microbiome to classify normal weight versus obese from microbiome data. And what you can see is that you can utilize the microbiome data, and you get a reasonable prediction here against BMI itself. You can see the area under the curve of 0.7 of predicting someone's BMI from that. This enhanced metabolic BMI that we just talked about on the last slide, the microbiome was in fact better at predicting it, which we think is the stronger health score for the reasons I just gave. And so you can see those kind of things as well. So again, a tight coupling between what you see in metabolites in the blood and what's happening in your microbiome. A final story that I want to say here about the microbiome is that it's also related to drugs. So we looked at statins. Statins were the most commonly used drugs in the cohort, so we just had a lot of data on them. And this was quite interesting, I think. So we just did a basic clustering into 4 separate groups, unsupervised, of the microbiome, and then evaluated what happened on the use of statins within those 4 groups. And what stood out was that one of those groups, this Bacteroides 2, as we called it here, so enriched in Bacteroides, in this cohort, people appeared to have double the lowering of LDL cholesterol in this microbiome than what we saw in the other microbiomes. So statins had a very weak, just detectable, but very weak effect on microbiome composition. But the microbiome composition appeared to have a strong effect on host responses to statins. And I don't have it here in this talk. I'll probably add it for the next one. I did just get someone that forwarded me a paper saying that there's been a clinical trial on this now in China, prospective, that they were able to show that in fact they could get differences in the response of statins by modulating the microbiome, citing this paper. So apparently this has been followed up on now, but I just got that. I haven't gone through it in detail yet. Here, this paper is in Med from Cell Press in 2022. But the other thing that's interesting in statins is that in addition to the lowering of LDL cholesterol, one of the downsides of statins is you do get a 9% increase in the rate of diabetes amongst people that use statins. And so what we saw here was that, again, when we looked at these different microbiome groups, in 2 of the groups, there's a very significant increase in markers of insulin resistance that you can see here, and in two groups, they're shown none of that. So it appears that the microbiome may be important both to the efficacy of the statin as well as to the side effect profile. Now, one of the things I think is interesting is that this is not cherry picked from a whole bunch of drugs we've looked at. This is just happens to be the first one we looked at. And I think, and there's been other studies. There's a great paper in Cell that showed that the microbiome on first approximation metabolizes away something like 13% of drugs. There's obviously a ton of microbiome drug interactions and that people are diving into now. And I think we're at the very early stages of really understanding all of these things. I think it's a pretty big iceberg. Okay, so that's what I'll say about the microbiome. We've also got a number that's been done on biological age on thousands of people, and I'll do this relatively quickly. I think we're all familiar probably with the concept of a biological age, or the age that your body says that you are, as opposed to the age the calendar says that you are. Lots of different ways to measure this. They don't all line up. You know, take that for what it's worth, but it's a tool to look uh, at what's there. I think what's most interesting here is to try to track how these change over time. And I have this caveat of if done properly, it can be tracked over time. I'll talk about what I mean by that here in a moment. And it can be based on different kinds of measurements, and they don't give the same results. On the residuals, in terms of like the delta age between chronologic age and a measured biological age. When we did this for proteomics derived, metabolomics derived, clinical lab derived, we saw a correlation of the residuals of 0.25. So signal, but not highly correlated. So here we see learning biological age for establishing a longitudinal action plan. So here, the thing that I wanted to point out is that we do use this Clamara-Duval algorithm. And I think this is a really important aspect, especially if you're going to use a biological age to track over time. The most important thing here is that it forces the slope of this line to be very close to 1. Right? And you do transformations to turn these into linear variables, orthogonal variables using PCA and so forth to achieve this. But basically, when we do this, you get something that's very close to 1. So that means you can use 1 year of biological age can be compared with 1 year of chronological age, because in a cohort, the algorithm is built so that the expectation is that you would see a 1 year per year increase. So that becomes important here. So this study that we published in the Journals of Gerontology in 2019, we wanted to see, if we thought this was relevant in some way, because you can always fit a curve, right? And you can always fit a curve, and you'll always have points that appear above the line and below the line, right, that have higher biological age or lower biological age. In and of itself, that's not meaningful at all. The question is that delta, does it tell you something, right? And so that's what we wanted to test here. And so here in the cohort, what we did is we looked at for all the different biological ages. And so these different colored lines here represent a biological age we did from the proteome, the metabolome, and from clinical labs. And so we were looking at the differences from different sources. But what I want you to notice here is that every place that there's a star, which is where we had statistically significant difference in the disease or condition as a function of that it materially moved the biological age. In every single one of these cases, the star is on the right side. So what that means is that in every single condition for which there was a statistically significant signal, it was associated with being higher on the biological age score. And we saw no examples of it being lower in biological age. So to me, this was an important test of, were we picking up something that was meaningful? And apparently, at least for health, it appears like the answer is yes. The other piece that I was actually more interested in was as people were going through a wellness program, and this is doing things that are, well validated for health, right? I mean, it's exercise and diet and movement and, get rid of junk food and, things like that. But as they're going through a health program, what we saw was that this estimated measure of biological age did in fact improve. So overall in the population, we saw a negative 0.16 reduction of 0.16 on biological age compared to the expectation that you'd go up by a year per year. Women were going down at a rate of 1.5 years per year on this measure. Men were getting better at a rate of 0.8 years per year on average, so going up by 0.2 instead of by a full year. So this showed two things then, that the biological age score being higher was associated with disease, and that if you went through programs aimed at improving your health, that you would in fact see it improve. And so we were heartened by that. We did end up turning one of those, the clinical lab-based version of this, or at least something similar to it, we ended up turning into the biological age test that we did at Thorne some years ago. Okay, so last section that I want to go through here, or second to last section, is around genomics and sort of what we can see from genomics and looking early. So one of the things that I think is really interesting is that genetics affects blood measures. So for example, LDL cholesterol. So we've just taken, so this is a case where we don't know anything about a person, what they eat, what they don't, and you just basically take your polygenic risk score against the actual measure of LDL, and you bucket it into 5 groups. And what you find is that there's this relationship between lower LDL and higher LDL. And at this point, we could capture about 12% of the variance. This is an older paper from 2019. Currently, you can capture more like 20% of the variance. So these have gotten a lot better. But anyway, I'm going to show the published version of this at the moment. So anyway, so this comes up. Now the question though is, should we treat people differently based on this genetics influence, or should we do what we do now, which is we ignore genetics and we just go by the LDL number against a standard clinical lab in Quest or LabCorp or wherever you get your LDL measured? So what we found when we took people again through the scientific wellness program that we were running at Airville at the time was that in fact it made a very big difference in terms of whether or not people would lower their LDL cholesterol for a lifestyle intervention versus not. And so what we saw was for the bottom 40%, where there was a significant gap, you could see lowering of LDL cholesterol, whereas in the top set, we saw no statistically significant difference in the ability to lower LDL cholesterol. So the basic idea here, which I think is a fairly general one, I mean, it'll depend on how much genetic prediction there is on any particular biomarker, but basically is this, that you can start with 2 individuals. They have, in this case, the same high level of LDL cholesterol. You have a genetic prediction that's different for them. The person that has the higher gap is the more likely to be able to see that move through lifestyle intervention. And so that delta represents an estimate of the non-genetic component of that value, which is more likely to be modifiable without hammering it, with a drug. You can get LDL, of course, to really come down by hammering it with a statin, for example. And so this is something that we're actually going to be deploying in a pretty big way right now. We're now working with the Emirati Genome Program in the United Arab Emirates, where they've sequenced 1 million people now. So almost all of the citizens of the country have actually had their genome sequenced now. And so we're actually getting to build this out for that population to do gap analysis like this across all these different biomarkers that are in their EHR systems against a population that has full genomics. So we're really excited about this, and we're pushing that forward in a big way with them, which is exciting. In the interest of time, I think I might, well, I'll mention this, I guess, just quickly. One of the other things you can look at is just what changes occur in people as they're going through a program like this. And one thing we had running the wellness program is that you do have people that transition into disease. And we had just a handful of people, but we had a few people, unfortunately, that got metastatic cancer while they were going through the program. And so we captured some data on that. This is an interesting example This is a known marker, CCAM5. It's a clinically used marker for metastasis. Um, what we found interesting, though, was that it appears like it may become elevated well in advance of what people had thought before. So here was a woman, 57, going through the program. At time point one, she already has very elevated CCAM5. She's not diagnosed at this point, but she comes into the program with very elevated CCAM5. By the way, we didn't know this in real time. The proteomics were actually run after the fact, just to be clear on that point. But time point two, time point three, elevated, time point four. So this is now 2 years of having elevated CCAM5. She then gets diagnosed with stage 4 pancreatic cancer and dies. Um, this is probably a signal. I mean, there's all the things that we could talk about in terms of, screening statistics, and we know, about that, but, you know, this is probably a signal. 69-year-old male, time point one, slightly, a little bit elevated here on CCAM5. Time point two, elevated. Time point three, elevated. Time point four jumps up, diagnosed with lung cancer, and dies. Woman, female, 57, starts low, jumps up. By the way, we did a statistical analysis. This level of jump is very unlikely, even though they're both in the normal range. This is very unusual degree of jump between the 6-month period. Time point three jumps to be the highest we ever measured. Breast cancer, stage 4, gets diagnosed. In her case, she got diagnosed at 3 and then transitioned to 4. So it's a very small N, but these were the only 3 people that got metastatic cancer, to our knowledge, in the program. We'll do a background distribution. So we did do the proteomics on the time series we had on these 3, and then a set of I don't know, 100, 150 different individuals that were matched from the population, and they were the only 3 that came up as elevated here in CCAM. So it's, early stage. It's small n up top. It's bigger n on the denominator. But it does show that there are likely lots of these kind of signals that come up quite in advance. Oh, this slide got messed up somehow when I copied it over, I guess. What this is supposed to say is that the greatest opportunity for AI isn't disease, it's wellness, which I'll argue. We've been building 2 systems that are called Healthspan Compass and Healthspan Synthesizer. I imagine these are aligned with, how a number of different groups are thinking about this in part. So this first system that we've built out of the lab does try to take a person's, and we've built this on top of all this scientific wellness data that I've been showing you through. So this is computing it up for one person and that went through the scientific wellness program that I mentioned. And so it's going through, and so the system will do things like, calculate their biological age, their polygenic risk scores, map their current state of health, compare them against the population, compare them against the genetic prediction for all their markers, and then gives advice about things that it thinks would make a difference in building out trajectory. So we're building out a lot around this in terms of just sophisticated systems for doing this, and we're hoping to make this live and release this out in the not too distant future. Hopefully first half of 2026 is where we're getting this done. And then the second that we're really working on is what we call Healthspan Synthesizer. And what this is basically, a virtual army of systems biologists that goes through and asks questions. So we've now got this operational on top of all the dense scientific wellness data. And so it allows researchers to come in and ask questions against the multi-omic data. It will generate an analysis plan, pull out the data, execute the plan, run the analysis, generate some figures, and write a paper about it. And so this is all the kind of thing that we know is emerging now in capabilities. There's all the tools that we're aware of. And I think just more and more of these are going to be coming available. And we've really shifted as a lab pretty strongly away from doing a lot of bioinformatics analysis to really building agentic AI systems that do classes of analysis. And so we're really trying to focus on that quite a lot. Um, Last slide here, longevity medicine, I think, is really coming into its own, thinking a lot about these early stages, like what does a risk manifestation stage look like, and how do we preserve health and enhance it? Transition phase, can we identify these earliest changes and tap them down? And then, of course, the disease manifestation phase, which is much better worked out and been the dominant focus of medicine for several decades. So with that, happy to take any questions. AI in health, I think, is a transformative opportunity. We went through a number of examples in the microbiome, biological aging, genetics, and so forth. But really, I think it's an incredible moment right now, and happy to take any questions. Thanks.
Thank you so much for this really great talk. Just to remind everybody that you can post your questions using the Q&A feature, and I see some folks have already posted questions. Maybe I'll start first. It was really fascinating to hear about the work you're doing with biological age. And I was wondering if biological age can be truly modifiable. Is there any concern about potentially promoting systems that over medicalize normal aging?
Yeah. So I get that question quite a bit in terms of like the over medicalization. I I tend to not think so much. It depends. I think it depends a lot on like how we think about interventions. Um, yeah, because the thing that I think is really important is that the earlier upstream that you go, like if you're working with someone that is asymptomatic, right? And you say, okay, I'm trying to lower my biological age. I'm not a huge proponent in that we should try to lower biological age using the kind of drugs that we use on late stage disease. You give chemotherapy to a cancer patient who might die. You would never give something like that to someone else. It is one of the reasons that I leaned more strongly into, well, certainly lifestyle, diet, exercise, things of that nature. It's also one areas that I've leaned a little more into like the supplement and natural product space than I did before. And one of the reasons for that is that if you're going to try to apply interventions in healthy individuals and you're going to try to kind of tap on different networks in different ways, you've got to have something that has an incredible safety profile. So it could be, certain drugs past that, right? It has to be something that's incredibly safe to use. A lot of the natural product space, at least the things that are like grass, generally regarded as safe and so forth, I think are other places to go. But you've got to titrate the kind of intervention you're doing to the stage that they're at. So I think that's kind of my response, I guess, on the over medicalization is we shouldn't do late stage medical things for health promotion, though. Those are different things.
Thank you. Earlier in your talk, when you were talking about the microbiome, there were a couple of questions. Do we know how GLP-1s are impacting the microbiome? And then also, are there any other strategies for optimizing personal microbiome health, like dietary strategies or other?
Yeah, there's excuse me. I'm not sure about the impact of GLP-1s on the microbiome, so I'll just put that out. I haven't actually looked at that in detail. I'm actually very interested in that. I need to go look at that more. I mean, calorie reduction goes massively, so there are big effects, but I don't have anything super specific to say about that right now. The opposite definitely is true that the composition of the microbiome can have an effect on GLP-1s. There are probiotics and things like this you can take to try to do that. Those have effects. They're not like the strength of the effects that the drugs have, but you can have modest effects there. In terms of the health of the microbiome, a lot of things, that I think people are aware of that you can do. Prebiotics tend to be better than probiotics in general. But combinations can be really good, kombucha, stuff like that people do, are healthy for the gut. I do take some pro and prebiotic mixes. I like Akkermansia, I think is one particular strain that's pretty important, a butyrate producer. I got measured as having low butyrate production a little while ago, so I've just got shifted onto some of those things. I'll retest in a couple months, see if it got better. Um, so I think some of those things are important. I take a pre and probiotic, a lot of days too, just to do it, try to deal with. And I've seen that it makes a difference for gut health issues, which I have periodically.
Um, and in the context of microbiome, there was a question if you've looked at or if you know of the relationship of microbiomes and mental predisposition, if any.
That there's there are definitely um, links there that make a difference. If you want to follow some of the science on this, like I serve on the advisory board for one of these NIH center grants that's led by Rima Kadura-Dauk at Duke, Rob Knight at UCSD, and Sarkis at Caltech. And there are it's kind of a huge mass of information that people are really trying to distill, but you do get strong predictive signatures related to Alzheimer's, related to Parkinson's, related to a number of different things. There is the well-known aspect that a lot of the neurotransmitters, serotonin in particular, really a lot of that is made in the gut. This is a little anecdotal, but was interesting. We had a number of us did some self-experimentation when I was at ISB, just taking resistant starch, which you can get as just unrefined potato flour. It costs like nothing, but you can get this, and supposedly it feeds the microbiome that makes, including the species that make serotonin. So we had, I don't know, about 10 of us that did it. It was interesting because for a couple of people, um, one of whom in particular really talked about he had never really dreamt much in his life. And he started taking this and like he just came and kept was raving to me. He's like, it's like I go to sleep, I'm like starting to have like these like technical, and I was like, yeah, that's dreaming, dude. Like I was like, like I felt, and I started taking mountains of it. It didn't make any difference to me. Leo took a bunch of it. And we couldn't see anything, but we had a couple people that tended to be, very soft spoken and, in different ways. And it was quite interesting to see, that, some of the things that people would say had happened. So that's a little bit anecdotal, but it was really interesting just to see kind of what came out of some of the self experimentation with some of those things that we saw.
Let's take one more question. I know we have so many questions. This has generated a lot of interest. There was one question about if you had one health span research project that you wanted to pursue without any limitations by funding, labor costs, or other practical limitations, what would it be?
I love that question. That's the kind of thing I would say to my wife is like, all I want is to be able to do all the science I want, unbounded by any financial constraints of any kind. Of course, doesn't exist in the world. The thing that I really want to do is it's what I put up on the screen at the end. I didn't say too much about it, which is this HealthSpan Horizons program. So that is kind of my blue sky element that I'm trying to do right now. So basically, I would like I think that what we want to do is just generate a massive, this won't be surprising, but a massive amount of like deep longitudinal data that we can apply for the study of health span. So I'm trying to set up that program so that we can bring in a lot of commercial data types, right? So if people are already, right, if they're using a Whoop or an Oura Ring or they're doing Function Health or Inside Tracker or like I'd like to have ways that people can like bring that with them, but then add on to it, and I'll raise some public money for this or philanthropy or whatever, but we'll do metabolomics and proteomics and lipidomics and all the stuff that we think about because with the advent of AI, and that's the point of those two AI systems I mentioned, Compass, so that people have a reason that they're getting something out of it, and then Synthesizer that will just sit on top of all that data and do nothing but try to write interesting papers about it all day, every day, right? I mean, you're kind of trying to say, can we do 100 postdoc years of effort on a data set every night, right? Can we do that? And so as we work towards that, and as we get the data to be deep enough, like I'll argue that the value of these really dense data where we just have many, many types of data on the same people and over time, the value of that has gone up exponentially now compared to when we started doing this a decade ago. Because of the AI systems, we can pull it in much stronger, we can do it much better. We can have these things like just go continuously, we can mine them like crazy, and we're going to learn so much about um, just unknown levers that exist. Like I showed some of them in just the data that we looked at. Like, wow, the microbiome looks like it has a huge effect on statins. Like, how would you know that? Or whether someone lowers their LDL cholesterol. Wow, the genome really predicts that. You never know that if you don't measure those things at the same time. And there'll be a ton of those. And in fact, if you look through the history of medicine, I was just having a fascinating conversation around this. So many things that we discovered were just pure serendipity, right? Someone just happened to notice. I mean, we all familiar with like penicillin's like the classic example we all know, but it's like there's many of these kind of things that happen. And what we can do with these systems now is just systematize that way more deeply. And so that's what I really want to, so if I'm unconstrained by resources and so forth, which I did have someone come to me, I was meeting with these philanthropists, and they were like, well, they wanted a plan for between $1 to $5 billion, which I wrote for them, but they haven't written me a check yet. But it's like, if we can do something unconstrained by money, um, it it would be that. I think if we could get hundreds of thousands, a million of people to do that over a decade, we would, with the right kind of AI systems attached to it, we would know 100 times more about what drives human health span than we do today.
Well, thank you so much for this wonderful presentation and discussion. I know we're running out of time and there's plenty of questions, so maybe you can contact Dr. Price individually for follow-up questions. I want to remind folks our next webinar is on February 12th with Dr. Huang. And I want to thank Dr. Price again and thank you all for attending.
Thank you, everyone.